Let us look at the phylogenetic tree of organisms that produce ice-binding protein. As we had seen, these are actually present in various type of microorganisms. Here are the kingdom, phylum, class, order and the family is given here. K, P, C, O, F are the ones. We have fish, bacteria, diatoms, plants, fungi, animals, copepods, insects. So all species are actually having this antifreeze proteins. So here are some of the different types. We have something called IBP with the beta solenoid fold alongside an alpha helix. These are some of the structures and we will be studying about these structures now. So they have packed polypropylene type coils, flattened silk-like solenoids, beta solenoid type AFPs. So the color, the blue color and the blue and red and the green color and the lectin-like type, these are all the different types of species which have different types of antifreeze proteins. So there are various categories of ice-binding protein. The major ones are called ice-nucleating proteins, antifreeze proteins, group 1, 2, 3, 4 and recrystallization inhibition proteins. These are all the three major categories. So what are the functions of them? The ice-nucleating proteins control the formation of ice crystals at high sub-zero temperatures and they contribute to the generation of ice crystals. So this is one thing which is different from the rest of the group. In the case of antifreeze proteins, either 1, 2, 3, 4, they decrease the freezing point of the body fluids to avoid freezing and they mitigate freezing around the organism's body fluids. And when we look at the recrystallization inhibition proteins, they inhibit recrystallization of ice in freeze-tolerant organisms. They have insignificant thermohysteresis activity. We will study about thermohysteresis in the later part. So these INPs trigger ice formation by lowering the nucleation barrier. So this is one unique thing they have. Whereas AFP's primary function is to prevent the ice formation by lowering the freezing point of water. In the case of recrystallization inhibition protein, they work on already formed ice crystals by inhibiting their growth or recrystallization to maintain smaller non-damaging ice crystals, because this is another type of mechanism by which they actually stop. One is they trigger ice formation but by lowering the nucleation barrier. The other one is they completely prevent ice formation. The other one, if the ice is formed, they will prevent the growth of the ice crystals. So that is how these three types of compounds behave differently to stop the ice growth. Let us look at some of the important features of these three class of compounds. When we look at the ice nucleating protein, they actually promote the formation of ice crystals, whereas antifreeze prevent ice formation. Recrystallization inhibition also inhibit recrystallization of the ice. So that means the ice should be already formed here. So what is the role in the organisms? The ice nucleating proteins initiate freezing to allow survival in icy environment, whereas the antifreeze proteins prevent freezing and protect vital functions. The recrystallization inhibition helps organisms survive through freeze tolerance. So we have seen freeze avoidance and freeze tolerance. So these actually do freeze tolerance. And when we look at the mechanism of action, ice nucleating proteins provide a surface for ice to nucleate, facilitating the initial formation of ice crystals, because they actually promote ice formation. But then how do they help the species survive? They actually have an effect on the freezing point. That is, they increase the likelihood of ice formation at higher temperature, that is sub-zero temperature also ice can be formed. They generally induce the formation of ice crystals in a supercooled water by providing a template for ice nucleation. So we will be looking at a little bit more about the ice nucleating proteins also. So on the antifreeze proteins, they bind to the ice surface and they disrupt the crystal growth and by causing thermal hysteresis, and they lower the freezing temperature. So this is a very unique thing and we will be focusing more on antifreeze proteins. And when we look at the recrystallization inhibition proteins, they bind to existing ice crystals, but they prevent their growth and the coalescence of smaller crystals thereby limiting the damage from large ice crystals because they prevent large ice crystal formation. And antifreeze proteins induce the formation of ice crystals in supercooled water by providing template for ice nucleation. They increase the likelihood of ice formation at higher temperature, that is sub-zero temperature also. So all of the three proteins increase the likelihood of ice formation at higher temperatures. But when we look at the ice nucleating protein, the structure is variable and often unique to species. They are active at high sub-zero temperatures. They are found in various cold adapted species. They generally lack a common structural motif. The variable can be hydrophobic or hydrophilic in nature. The structure of these compounds, these proteins can be either hydrophilic or hydrophobic. They are diverse and they may not share a common ancestry. So this is the uniqueness of ice nucleating proteins. When we look at antifreeze proteins, there is a consistent common domain, but they do have diverse structures. They function across a range of sub-zero temperatures. Fish, plant, insect and some microorganisms produce antifreeze proteins. The right-handed beta helical structure is a common domain in all these AFPs. They are typically hydrophobic and that's very crucial for ice binding. Threonine and alanine are critical residues which are involved in the structure. They are structurally diverse and not genetically related. And when we look at the recrystallization inhibition proteins, they are less diverse and they primarily focus on inhibition only. They operate in freeze-tolerant organisms like fish, plant and insect. They have simpler or more common uniform structures across species. They are generally hydrophobic, less focus on ice binding. Their involvement varies and they are less well defined. They are less diverse, may have some genetic similarities. Let us look at the different types of species in which these compounds are actually present. We have insect and fish, bacteria, plants, cryophilic nematodes. There are few features which are very unique to different types of species. The thermal hysteresis is very high in the case of insect and fish because they have AFPs. Bacteria, it is adequate, but in the case of plants it is very low, and cryophilic nematodes does not have this thermal hysteresis. When we look at ice recrystallization in the insect and fish, it is very low, and bacteria and microorganisms do have something, but the plant has this one very unique. So if you look at the insect, fish and the plants, thermal hysteresis is a very important feature in the case of insect and fish, whereas for the plant it is the ice recrystallization inhibition. So that's how we can easily differentiate. Each species have a different type of process to prevent the ice growth. And when we look at the AFP secretion, all these things have AFP secreted. In the case of insect and fish, it helps in the survival, whereas in the case of bacteria and microorganism, it aids in the nutrient and oxygen gathering. In the case of plant, it supports nutrient acquisition, and in the case of cryophilic one, it maintains the small ice crystals. The example of the species are given here. And the lethal temperature threshold for insect and fish is generally above -3 degrees centigrade. Bacteria and microorganisms can endure lower temperatures. Plants can survive up to -30 degree centigrade, and cryophilic nematodes can survive up to -16 degrees centigrade.